We look now at the key aspect of DNA analysis by the forensic chemist, tied closely to the uniqueness of a person's DNA. You're used to hearing that every single person in the world has a unique coding on their DNA, and this is true. But when it comes to forensic chemistry, we're interested in parts of that unique code of DNA. Let's have a closer look. Remember, 99.9% of our coding DNA is identical, so it's not much use in distinguishing between subjects. We've already discussed that if we looked at the genes, the coding regions in every human being, we would find that we have the same coding regions, and that's why we're of the same species. We are what our DNA codes for, if you like. So it's no use looking at those parts of the DNA code, because all it would tell us is that we have a human being of which there are 6.5 billion subjects in a criminal investigation. The non-coding sequences, or junk DNA, is present in all DNA molecules as well. It's these areas of the DNA strand that are different between organisms. What we can do is examine the length of these segments of non-coding DNA, and by doing that, we are able to identify a suspect with an error chance of less than 1 in 10 billion. What you can see here is an example of an electrophoresis gel that's showing the banding patterns when we're looking at junk DNA. But let's take a closer look at how it actually happens. If you have a look at this schematic representation that comes from your textbook, the upper strand is an example of a portion of DNA that may have come from, say, person A, suspect A. Down below that is a portion of the same section of DNA that comes from person B. We've schematically represented two areas on the DNA strand. One is coloured red and one is coloured green. What you can see is that the red bits are of exactly the same length, though they tend to be sitting on different areas or loci of the strand. So take a look at the first red one up the top from person A. You can see that there's a corresponding red band on person B. Slightly different position, but exactly the same length. Same for the second red band, same for the third, fourth, fifth, sixth, and so on. We will refer to these red areas as exons. The green sections on the strands of DNA are of different lengths. If you take a look at the leftmost green strand on person A, you'll see that it's of a certain length, say about 1.5 centimeters in length. But the first green strand on person B is much smaller, only a few millimeters. These are examples of what we call introns. They are very different between individuals, and it's these areas that give people their individual DNA profile. Exons are the coding regions that contain genes for cell function. Introns have no known function, and they vary greatly and are often referred to as junk DNA. Well, how do we remember the difference between these two? I remember it by thinking that exons are excellent. We know what they do. They code for cell function. That's where we have genes, and they're the same in all people, so that's excellent. When we're talking about DNA, the excellent part are the exons, because that's what makes sense and gives rise to cell function. Introns are of interest in forensics. Even though we don't know what they do, because they're different, they are going to be the distinguishing feature of DNA, so introns are of interest to us. So, just in brief, exons, they're identical within the human race, no matter who you are, because they code for human characteristics. Introns are different between every individual human being, and therefore they're of great interest to us. What we can do is we can examine these introns to establish what's called a familial link. Familial looks like the word family, so we can establish that somebody is related to somebody else, has a familial link, by examining certain parts of the DNA strand that contain specific introns. Let's have a look. Parents contribute 50% of their DNA to their children. Now, it's not the same 50% to each child, but just think about it like this. Sex cells in the human body contain half the normal amount of DNA as every other cell. These are called haploid cells. We don't go into this in any depth at all. It's a fascinating process called meiosis, where we get the replication of DNA and the passing on of DNA coding. But for our intents and purposes in forensic chemistry, it's sufficient to recognize that children have received half of their DNA from mum and half of their DNA from dad. In other words, parents contribute 50% of who they are, DNA-wise, to their children. That's why children have some characteristics that are similar to mum and some characteristics that are similar to dad. Children, therefore, will also have half of their introns identical to their mother and half will be identical to their father. Now, to establish familial ties, relationships within families, this might happen in a custody battle or to force an estranged father perhaps to make family payments. A certain set of introns is what we call amplified via a process called PCR and then separated with electrophoresis. PCR is examined more closely in the Moodle lesson. And so what we do is we have a look for the presence of introns and we amplify them. In other words, we magnify how many of those we have, then we run them on a gel and have a look at the banding patterns. Now, a child will have half of their introns common with their mother and half common with their father. Siblings, so brothers and sisters, will have 50% of their introns the same. Cousins have 25% of their introns the same. So you can establish if you have a long-lost sister or brother, because 50% of your introns will be identical to theirs. Likewise, you can find a long-lost cousin by doing some DNA profiling. If you have 25% or a quarter of your DNA introns are the same, you could conclude that they are indeed your cousin. Remember, this is very interesting as we examine the introns. We call this examination of the DNA introns DNA profiling because we're forming a profile for an individual. This is how we do DNA profiling. Once the sample of DNA has undergone amplification via PCR, check Moodle again if you're not sure of this, and the specific introns have been cut and amplified, we run them on a gel. Now remember, the longer the intron is, in other words, the length of the strand, the slower it's going to move through the electrophoresis gel. The electrophoresis gel is like a little matrix at a microscopic level, so of course if you have a bigger molecule, it's harder for it to slide through the gel, so it takes longer to move toward the electrode. If the intron is shorter, it will move faster through the gel towards the electrode. And so remember now, we're going to separate sections of DNA called introns based on the length or size of that section of the DNA. Here's again that picture of a DNA profile. Imagine now you can see the index finger up the top right and you can see some faint wells along the top of the gel. The DNA that has been amplified via PCR would be placed in those wells and then the current would be turned on. The banding pattern shows how introns of different lengths have moved through the gel at different speeds. We have two banding patterns set up on this particular gel, and so halfway down the gel you can see again another set of wells and banding patterns continuing on down the gel. Now the intensity of that light under the ultraviolet spectrum reflecting out of those banding patterns shows us the concentrations of those numbers of introns and how far they've travelled. And so we are able to profile somebody's DNA by looking at how the introns spread out over time. If this was the DNA profile, say, of a mother, and we needed to establish if she had a child, we would then run another gel with the child's banding patterns, and you could place one gel over the top of the other, and if half of the banding patterns matched exactly this particular gel, you could conclude that this subject was indeed the child of the mother. So there's an example of DNA profiling We're very interested in the introns. They are of interest to us in forensic chemistry. They are the non-coding parts of the DNA strand that is different for every individual on the planet.